Assalamualaikum dan salam sejahtera. This is class for General Biology. I'm Dr. Shaida and I will be your instructor for this course. We will continue our lesson on Chapter 2, Cell Division and Reproduction under the subtopic of Meiosis. In the previous lesson, we have learned about development of somatic cells. The somatic cells undergoes the mitosis process as proliferation process to produce new cells. The result of mitosis is two identical daughter cells, genetically identical to the original cells, all having two N chromosomes or diploid cells. If the somatic cell undergoes the mitosis process for its development, how about the gamete cells? As recap from the previous lesson, a gamete is a reproductive cells of an animal or plant. In animals, female gametes are called as ovum or egg cells, and male gametes are called sperm. Ovum and sperm are haploid cells with each cell carrying only one copy of each chromosomes. Haploid describes a cells that contain a single set of chromosomes. The term haploid can also refer to the number of chromosomes in egg or sperm cells, which are also called as gametes. In humans, gametes are haploid cells that contain 23 chromosomes, each of which as one of a chromosome pair that exists in diploid cells. The number of chromosomes in a single set is represented as N, which is called as haploid numbers. In human, we always refer to the N equal to 23. Germ cells are located in the ovaries and testes and develop into gametes. Gametes are the egg and sperm cells in your bodies that contain DNA can be passed to the offspring. To form a new progeny, an egg from the mother and sperm from the father will fuse together. The egg and sperm each have haploid cells will form a full diploid cell sets of chromosomes. The egg and sperm together will give the baby the full set of diploid chromosomes. So, half of the baby's DNA comes from the mother and half comes from the father. Inside each human cell, DNA is tightly wrapped together in a structure called as chromosomes. Every normal cell has 23 pairs of chromosomes for a total of 46. 22 pairs of the chromosomes are the same in male and female. These are called as autosomes. The 23rd pair of the chromosome is known as the sex chromosomes that determine the sex of the baby. Female have 2 X chromosomes and male have 1 X chromosome and 1 Y chromosome. Diploid cells have 2 sets of chromosomes that are inherited from mother and father cells. Organisms have a specific stable number of chromosomes in their cells. For example, humans have 46 chromosomes in each diploid cells. If gametes were also diploid, the number of chromosomes in each generation will double within each fertilization event, leading to unmanageable increase in chromosome number over successive generations. How can the diploid cells of humans turn to be half? The answer is by meiosis. The haploid cells of gametes are produced during meiosis, which is a type of cell division that reduces the number of chromosomes in a parent diploid cell by half. It is produced during sexual reproduction. Being haploid means they can only contain one set of chromosomes. The first process of meiosis was first described in the mid of 1870s by Oscar Hadwood, who have observed it while working with the sea urchin eggs. Later, Edward von Veneden expanded upon Hedwig's descriptions, adding his observation about the movement of the individual chromosome within the germ cells. By definition, meiosis is the division of special kinds of cell, and it also can be called as sexual reproduction. All animal cells conduct the meiosis process. Similarly, seaweed, fungi, and plants conduct meiosis too. For plant, it can be produced either sexually or asexually. When new plants are produced from existing plant parts like pieces of leaf, stem, or root, reproduction is by asexually, and the only type of cell division that has taken place is by mitosis, where one diploid cell produces two identical diploid cells. If new plants are instead produced from seeds, this is a strong indication that it is produced bisexually. The meiotic cell division is required to initiate the haploid stage of the plant that ultimately results in production of eggs and sperm cells central to the sexual reproduction. The sexual reproduction is mainly happen in angiosperm. Angiosperm is also called as the flowering plant. There are about 300,000 species of flowering plants, which is the largest and most diverse group within the kingdom of Plantae. Flowering plants reproduce sexually through a process called pollination. Flowers contain male sex organ called stamens and female sex organ called pistils. The anther is the part of the stamen that contains pollen. Pollen contains the male gametes. Plant growth is divided into two generations that are diploid 2N and haploid 1N. Higher plants or angiosperm have a long-lived sporophytic generation that is the diploid sporophyte. The sporophyte is the growth that you would easily recognize as a plant. Through the process of meiosis, the sporophyte produces haploid spores in the flower. The spores are the gametophyte generation. Flowering plants undergo meiosis. Meiosis occurs in the male flower parts to produce pollen and the female floral organ to produce egg cells. When pollen is released from the flower, the female gametes are hidden from direct view in the ovary. Spores grow by mitosis, producing more haploid cells. This is the gametophytic generation. When the haploid gametes, where the male pollen and the female egg cells unite from the pollination and fertilization process, they will reform the sporophytic generation, producing a diploid zygote or 2N. The zygote grows into the embryo of the seed and eventually into the plant that we can see. The process of meiosis begins similarly to the mitosis in that a cell has grown large enough to divide and has replicated its chromosomes. However, meiosis requires two rounds of division. In the first round, it is known as meiosis 1. Then, it gets replicated and form homologous chromosomes to be segregated. During the meiosis 2, the sister chromatids will then segregate. Meiosis 1 is called a reductional division because it reduces the numbers of chromosomes inherited in each of the daughter cells, in which the parent cell, which is 2N, while the daughter cell will only have 1N. Meiosis 1 is further divided into prophase 1, metaphase 1, anaphase 1, and telophase 1. Metaphase 1, anaphase 1, and telophase 1, which are roughly similar to the corresponding stages of mitosis, except except that in prophase 1 and metaphase 1, the homologous chromosomes pair out with each other or synapse and are called bivalent. Crossover events can be seen as chiasmata on the synapse chromosome in late meiosis 1. Crossover function to hold the homologous chromosome together during meiosis 1 so they can orient correctly and segregate successfully. Crossing over also reshuffle the allele combination along the chromosome, resulting in genetic diversity that can be selected in a population over time. Just like the mitosis, meiosis also consists of preparatory phase called as interphase. At the beginning of the first meiotic division, the nucleus of dividing cells starts to increase in size by absorbing the water from the cytoplasm, and the nuclear volume increases about 3 folds. Meiosis is preceded by an interphase 1 which is nearly identical to the interphase preceding mitosis. During the interphase, the DNA of the chromosome is replicated during the S phase. After DNA replication, each chromosome becomes composed of 2 identical copies, which is called as sister chromatids that are held together at the centromere until they are pulled apart during meiosis 2. The interphase is characterized by the following features. First, the nuclear envelope remains intact, and the chromosome occur in the form of diffuse, long coil and indistinctly visible chromatin fibers. Second, the DNA of the chromosome is replicated during the S phase. Third, the DNA becomes double. After DNA replication, each chromosome becomes composed of 2 identical copies that is called as sister chromatids that are held together at the centromere until they are pulled apart during meiosis 2. And number 4, due to the accumulation of ribosomal RNA and ribosomal protein in the nucleolus, the size of the nucleolus is significantly increased. And if it is in animal cells, a daughter pair of centrioles originates near the already existing centriole, and thus an interphase cell has 2 pairs of centrioles. And in the G2 phase of the interphase, there is a decisive changes that direct the cell toward the meiosis instead of mitosis. There are 4 subdivisions of meiosis 1, which are the prophase 1, metaphase 1, anaphase 1, and telophase 1. During prophase 1, the homologous chromosomes pair together and form a synaptonymal complex. Crossing over occurs within the synaptonymal complex. A crossover is a place where DNA repair enzymes break the DNA of 2 non-sister chromatids in similar locations and then covalently reattach to non-sister chromatids together to create a crossover between non-sister chromatids. Next, it goes to the metaphase 1. In this stage, tetrads line up on the equator of the metaphase plate, and each chromosomes of homologous pair attached to the spindle fibers from opposite end of the poles, in which sister chromatids attach to the fiber from the same pole. Then it enters the anaphase 1. During this phase, chiasmata dissolve and homologous chromosomes move to the opposite poles. The homologous are pulled apart and move apart to the opposite end of the cells. The sister chromatids of each chromosomes, however, remain attached to one another and don't come apart. Finally, in telophase 1, the chromosome arrive at the opposite pole of the cells. The nuclear envelope reforms and the resulting cells have half of the number of the chromosome, each consisting of two sister chromatids. Cytokinesis usually occurs at the same time as telophase 1, forming two haploid daughter cells. Interkinesis or cytokinesis is similar to the interphase except no chromosome duplication occurs. Daughter nuclei become enclosed into the separated daughter cells. At this stage, each daughter cell is haploid and has only one set of chromosomes or half of the total number of the chromosome of the original cells. Cells move from meiosis I to meiosis II without copying their DNA. Meiosis II is a shorter and simpler process than meiosis I. At the completion of meiosis I, there are two cells, each with one replicated copy of each chromosome or 1N. Because of the number of chromosomes per cell has decreased from 2 to become 1, meiosis I is called as reductional cells division. Meiosis II resembles the mitosis, with one sister chromatids from each chromosome separating to produce two daughter cells. Because meiosis II, like mitosis, it results in the segregation of sister chromatids. Meiosis II is called as equational divisions. Meiosis II may begin with the interkinesis or interphase II. This differs from interphase I, that is, no S phase occurs as the DNA has already been replicated. Thus, only a G phase occurs. Meiosis II is equational division as the cells begin as haploid cells and end as haploid cells. During prophase II, chromosome condense. The centrole moves towards the poles and the nuclear envelope disintegrates. As the centromeres move apart, the spindle forms between them and the spindle microtubule begin to capture the chromosomes. During metaphase II, the sister chromatids are maximally condensed and aligned at the equator or the metaphase split of the cells. Besides, the sister chromatids attach to the spindle fibers from opposite poles. In anaphase II, the centromeres divide. The sister chromatids separate and are pulled towards opposite poles of the cells. In telophase II, the chromosome begin to uncoil, and the nuclear envelope and nucleoli begin to reform around each set of chromosomes, and the chromosome decondense. Followed by cytokinesis, the telophase II marks the end of meiosis. 4 haploid daughter cells are formed as a result. Cytokinesis separates the two cells into 4 unique haploid cells. At this point, the newly formed nuclei are both haploid. The cells produced are genetically unique because of the random assortment of paternal and maternal homologous and because of the recombining of maternal and paternal segments of the chromosomes that occur during the crossover process. In summary, meiosis is a specialized type of cell division that occurs in sexually reproducing organisms. Unlike mitosis, which produces 2 identical daughter cells, meiosis produces 4 non-identical daughter cells, each with half of the number of chromosomes as the parent cells. The cells goes through similar stages and uses similar strategies to organize and separate chromosomes. It is accomplished in meiosis using two-step division process. Homologous pairs are separated during the first round of cell division called as meiosis I, while the sister chromatids are separated during the second round called as meiosis II. Since cell division occurs twice during meiosis, one starting cell can produce 4 gametes in form of eggs or sperm. In each round of cell division, cell will go through 4 stages by prophase, metaphase, anaphase, and telophase. Meiosis is a crucial process in sexual reproduction that ensures genetic diversity among offsprings. That's all for today's class. Thank you for listening. See you in the next lesson. Bye bye. Assalamualaikum warahmatullahi wabarakatuh.